So now that we've talked about the main components of the cell membrane, let's talk about how we get things in and out through that membrane. We're going to be looking at 3 forms of transport: passive transport, active transport, and bulk transport. For this video, we're going to focus on passive transport, which can break down into diffusion and osmosis, and diffusion breaking down to simple and facilitated. So when we're looking at a solution, it's made up of a solvent, which is going to serve as the liquid that we dissolve something in, and the dissolved substances themselves called the solute. So the solvent, let's say we're looking at salt water. The solvent would be the water, and the solute would be the salt. Solutions can be solids, liquids, or gases. So while we're going to kind of focus on a liquid, just because that's a little bit of an easier to understand example, they can be solids or gases as well. Now, we're focused on passive transport for this video, and passive transport does not require energy. It occurs spontaneously, meaning it's just going to happen regardless of what the input is, right? If you leave something completely alone, passive transport can happen, no energy input needed. So for cells, this means free, right? We don't have to spend any ATP or energy in order to allow passive transport to happen. And being as efficient as possible is in the cell's best interest. So passive transport is actually pretty useful to our cells. Now, the movement of passive transport, because it doesn't require energy and it happens spontaneously, means we're only going to be able to move in one direction. Not necessarily a direction in terms of in or out of the cell, but in terms of the concentration gradient. Passive transport moves molecules with the concentration gradient, meaning we're going to go from a high concentration towards a low concentration until we reach equilibrium, until both sides of the membrane or whatever we're looking at, until that concentration has equalized across the entire area. So let's look at diffusion, which is kind of a general passive transport form, and we'll break it down to simple and facilitated. So diffusion is the movement of molecules from areas of a high concentration to a low concentration. That's the basic. It doesn't matter what we're looking at, what kind of molecule. We're just looking at generic molecules going from high to low. Simple diffusion is where we pass these molecules right through the membrane. We don't need any sort of special channels. We don't need anything to kind of help us along. So these are the molecules that can push their way past the phospholipids, either towards the inside or the outside of the cell. Facilitated diffusion is you need a little bit of assistance, right? We need something to facilitate this process. And this can be done by protein channels that serve as openings for larger molecules. A good example is something like water. Water cannot necessarily push its way past the phospholipids because water is polar. But cells have these openings, these channels called aquaporins, that will facilitate the movement of water. So let's look at diffusion. And I know I demonstrated this in class, but if you don't remember or if you couldn't quite see what I was doing, what we did in class in lab was we took a beaker of water and we dropped some coloring, some stain in that water. And this is something, if you want to kind of get your mind around it with a visual, you can do yourself with some water and food coloring that you might have in your pantry. So you take a cup of water and you take your food coloring, you put a few drops into the water. Now, the food coloring you're dropping in is highly concentrated. There's a lot of that coloring molecules packed into a single drop. But as soon as you drop the food coloring into the water, the drop does not stay together. It starts to disperse out. So we've dropped it into a solvent, and immediately the molecules are randomly moving around, bumping into each other and spreading out away from each other, dispersing themselves evenly, or at least working towards dispersing themselves evenly throughout the entire glass of water. So that random movement of the molecules from the food coloring is going to cause them to evenly distribute throughout the entire glass of water. So if you try this at home, you have your cup of water, you drop a few drops of food coloring in it, and you let it sit without touching it, that food coloring will evenly disperse throughout the entire cup of water given enough time. That's diffusion. And it's a good example of kind of what we're talking about with simple diffusion, where we're just passing through and it's happening naturally and we don't need anything to assist that process. Right? We see that here where we see like oxygen, right, and carbon dioxide. These guys are small enough and don't have any polarity to them where these molecules can pass right past the phospholipids. So oxygen can pass through in one direction and carbon dioxide can pass through the other. This is something we see in like the lungs when we look at the alveoli that control the intake of oxygen and pushing out the carbon dioxide. They're happening because there's a higher concentration of oxygen on one side of the alveoli and a higher concentration of carbon dioxide on the opposite side. And so oxygen and carbon dioxide can easily diffuse across the membrane without any sort of help. For things that are bigger, right, let's say we're looking at glucose or we're looking at some sort of larger molecule that maybe has a charge or maybe is polar or is just simply big, we need a channel. And so here's this protein that's going to serve kind of as a doorway, a special doorway for that protein, that molecule, for that molecule to pass through. It's still passive transport because we don't require energy input. All we're doing is providing an opening that this molecule can pass through. But we're still moving from a high concentration to a low concentration, and we're still not spending any energy to do it. Osmosis is a special form of passive transport where we're focused on water. So osmosis is the passive movement of water through a membrane. Now, when we're talking about the movement of water, we tend to, and this is where the language can get a little confusing, because water is still going to want to move from that high to low water concentration. But when we're talking about the movement of water, we're usually putting it in the framework of the solute concentration, not the water concentration. So even though this is passive, it's going to sound like active just because we're moving from low to high in terms of the solute concentration. So water is going to move from the low solute concentration area to the high solute concentration area, right? And it depends on what is dissolved in with the water, right? The goal of water is to balance the solute concentrations. This is especially important if the solutes are not able to move across the membrane. So let's say it's a charged ion on either side and we're not able to move that ion past the membrane, water can move. So since the solutes are locked away, maybe there's a high concentration of a solute on the outside of the cell and a low concentration of the solute on the inside, and the solutes are locked, water is going to move to the high solute concentration outside of the cell so that we can achieve equilibrium for that solute concentration. So we're basically just using water to do the dilution of the higher concentration of the solute, since the solutes themselves cannot move. Right. The relationship between the solute concentrations on the inside versus the outside of the cell is what we call tonicity. Right. So what does that solute concentration look like in comparison to the inside of our cell? So we're going to look at some animal cells and plant cells and how they respond to different types of solutions and what those solutions can look like. So let's start with the easiest one, which is the isotonic solution. Iso tells us same. So the isotonic solution, the concentration of solutes is equal both inside and outside the cell. Right? It's the same. And so that means that the concentration inside this red blood cell from an animal is going to be the same as the concentration around it. And same thing with the plant cell. Right? So concentration of solutes here and concentration of solutes here are the same. Now, what does that mean for water? It means that water can move in and out, but it's not going to go in any sort of direction. It's a net zero. Think of it like if you go to a store and they only allow 100 customers in the store at a time. Right? You're waiting in line to get in. You're not allowed in until someone leaves. Someone walks out, you walk in. The number of customers in the store is still the same, even though the individual customers may have moved around. In a hypertonic solution, hyper means above. Like if you're hyperactive, you have a ton of energy. And so the solute concentration is higher outside of the cell. So let's say we're looking at salt water. So there'd be like a lot of salt dissolved in the water outside of the cell and very little salt on the inside of the cell. What does that mean for our animal cells and plant cells? It means that water, because it wants to move to where the high solute concentration is going to get pulled out of the cells and it's going to dehydrate them. A hypotonic solution, hypo meaning below, means the solute concentration is higher on the inside of the cell and lower on the outside. This means water is going to move towards the high solute concentration inside of the cell. Now, this is where animals and plant cells differ. Animal cells only have a plasma membrane, and so when water gets pulled inside the cell and it starts overly inflating that cell with water, it's kind of like if you overfill a water balloon. Eventually, that water balloon is going to weaken and burst open. So the animal cell has no protection against a hypotonic solution. They're just going to keep taking on water until they finally burst. The plant cell, however, has a cell wall in addition to their plasma membrane. And so they're able to actually counter that high pressure. And plant cells actually do kind of like a hypotonic solution because they can pull in that water and remain what we call turgid, where they have the high osmotic pressure inside of the cell and the cell wall pushing back. And so this is something, if you wanted to, you could practice in your own kitchen, in your fridge. If you get something like celery or asparagus, some sort of vegetable like that, and you just leave it in your fridge, you'll notice that it wilts, and it wilts pretty fast. But if you want to keep it more crisp, what you could do is place that celery or asparagus or whatever it is in distilled water, just pure water. Because that water has so little dissolved molecules in it, compared to the celery cells themselves, the celery cells are going to actually take that water from the cup you put it in into the cells. And this keeps the celery nice and crisp. So water gets pulled into the plant cells because the concentration of solutes is higher inside the cell compared to the outside. This can keep your vegetables like that fresh for a longer period of time. If you were to do the opposite though, you were to take your celery and put it in some salt water, which I'm not sure why you would, but you could always try it. If you did that, the high concentration of solutes is outside the cell. That's going to sap that water. It's going to pull that water out of the cells into the cup. If it does that, your celery is going to wilt and shrivel up, even faster than if you just leave it alone in your fridge.